Okay, starting webinar. I'm going to wait until it's around the 60 people mark, and then I will put on the slide. All right, the webinar is live.
Good afternoon, everyone, and welcome to the first UCLA CTSI Distinguished Speakers Series seminar for the 2021-22 academic year. My name is Mike Teitel, and I have the great privilege of introducing today's kickoff seminar speaker, one of our own, Dr. Lili Yang. Dr. Yang received a Ph.D. in biology and was a postdoctoral fellow with Dr. David Baltimore at Caltech before she moved as an assistant professor to UCLA in 2013. Here at UCLA, she rose through the ranks to her current position as an associate professor in the Department of Microbiology, Immunology, and Molecular Genetics. Dr. Yang is also a member of the UCLA Johnson Comprehensive Cancer Center and a member of the David Geffen School of Medicine Cancer Research Theme. Dr. Yang and her research group study tumor immunology and cancer immunotherapy with a special emphasis on developing gene and cell-based immunotherapy for cancer. Results from her work include over 35 publications, 15 patents, 2 active clinical trials, and 2 biotechnology startup companies. Dr. Yang has also received multiple prestigious awards, too many to include. One would be the Innovators Under 35 Award from the MIT Technology Review magazine. Another would be the NIH Director's New Innovator Award. Yet another would be the Young Investigator Award from the American Association of Immunologists. And the last one I'll mention is an Outstanding New Investigator Award from the American Society of Gene and Cell Therapy. Clearly, Dr. Yang is quite accomplished and doing great things. Now, during Lili's virtual seminar, if you have any questions, please use the chat function at the bottom of the screen. These questions will be answered following Dr. Yang's seminar, which today is entitled Targeting Monoamine Oxidase A for Cancer Immunotherapy. Lili, it's a real great pleasure to have you kick off the CTSI series for 2021-2022. All yours.
Thank you very much, Mike, for this very kind introduction. It's a great honor, to be the first speaker at your new series. Today, I'm going to talk about some new progress from my lab on the cancer immunotherapy. A little background about cancer immunotherapy. In the past 2 decades, it has achieved significant progress in this area. In fact, now it's becoming the 5th pillar for standard cancer care, joining the family with radiotherapy, chemotherapy, surgery, and precision therapy. Cancer immunotherapy works through harnesses and enhances the body's self-defense system to battle cancer. One good example is called the immune checkpoint blockade therapy, ICB therapy. What it does is that it targets those key molecules, works as negative regulator of the immune reaction against the cancer. And by using monoclonal antibodies like anti-CTLA4 or anti-PD1, so those monoclonal antibody block those negatively regulatory molecule and therefore release the brake for the immune system to fight cancer. Those two ICB therapy has been approved by FDA for treating over 10 different malignancies and has revolutionized the treatment for many of those cancers. However, despite the success, only a fraction of patients with cancer respond to those two therapies, and the most responded suffer tumor recurrence. One of the underlying mechanisms can be the immune evasion mechanism, where cancer mutated away of those molecules that can subject to those therapies. So nowadays, a very hot area in cancer immunotherapy is try to identify new immune checkpoints and develop a new combination treatment, like to trying to cover more cancer type, more cancer patients, and to try to prevent the possible tumor immune evasion. And my lab is also have a major research direction aligned on this area. Today, I'm very happy to share with you some of our new progresses along this direction, where we find a molecule called the monoamine oxidase A play an important role on regulating the immune response to cancer. And interestingly, this single molecule not only regulates one single type of immune cells. Recently, we published this two work, like reporting, like Mao A regulate both T cell mediated anti-tumor response and also the tumor-associated macrophage mediated anti-tumor response. Those work are led by two talented postdocs working in my lab, Shirley Wan and Ryan Wan. Due to the time limit, so today we probably will mainly focus on talking about the T cell side of the story, but I will also touch the macrophage side of the story. So since we find out those this Mao A molecule is important for regulating anti-tumor immunity, we also spend part of the research trying to explore this new target to see whether we can, based on this finding, develop a new version of the cancer immunotherapy. I'm going to share with you this work now. So I'll start with the why question, right? Why we study this Mao A molecule, okay? So it started by our finding that Mao A gene is upregulated in tumor infiltrating immune cells. Here is how we perform our screening study to search for the possible new immune checkpoint in molecules, like beside CTLA4, beside the PD1, right? We're trying to look at this, whether that's a newer target that we can explore to expand the existing ICB therapy. What we did is that we use a preclinical animal model. Specifically, we use the preclinical mouse tumor models. We grow tumor in mice. We isolate the tumor infiltrating immune cells from those animals, and we study the gene expression profiles of those immune cells. So surprisingly, beside the typical molecule that classified as immune regulatory molecule, like CTLA4, like PD1, we also find there's another group of molecule which regularly considered to be molecules regulate the neuronal activity, also get upregulated in tumor infiltrating immune cells. And that triggers our interest. So what is Mao A? What people know about this molecule? Okay, so Mao A gene encodes this monoamine oxidase A. Mao A, it's an enzyme that's located on the outer membrane of mitochondria. And its known function is to break down monoamine substrate, including those most best known as neuronal transmitter, such as serotonin, okay? And non-surprisingly, actually, the best known function of Mao A is in the brain. So people know that in the brain, Mao A expressed by the neurons, okay? So what it regulates over in the brain and between the neuronal synapse is that it controls the metabolism of serotonin. So by break down serotonin, Mao A regulates the availability of serotonin to stimulate the neuronal transmission. Okay, so in fact, like it play an important role through regulating serotonin to regulate the human mood and behavior. And in the history, like people find that a population of people like contain some mutation of the Mao A gene is associated with certain behavior change. In fact, associated with more aggressive, even violent behavior. That is why if you search on the internet and back in time, that Mao A has gained the nickname called the warrior gene because it controls the aggressiveness of the behavior, okay? So because the known function of Mao A in brain and its ability to regulate mood and behavior, it's already explored as a drug target. And a small molecule Mao inhibitors called Mao As have been developed and are clinically used for treating depression and other neurological disorders. In fact, Mao As is the first major group of antidepressants to be approved for treating depression. So knowing about this, we are curious as we know that Mao A routinely work in brain, regulate the neuronal activity. Now we find this gene also expressed in the immune cell, especially for the immune cell infiltrate tumor and they upregulate Mao A. What does this mean, right? So our question is that whether it's indeed plays some physiological role regulating the anti-tumor behavior of those tumor-infiltrating immune cells. So as an immunologist, we use some typical research tool help us to address this question. And the first tool we use is the Mao A deficient mice. So we use those mice, then we challenge the mice with tumor. We ask whether in the absence of the Mao A regulation, whether we can see some changed anti-tumor immune reactions. So here is what we did. So we compare the Mao A knockout mice with the Mao A wild type mice. We challenge those mice with two kinds of tumor model. One is the MC38 colon cancer model. Another is the B16 ova melanoma model. Both are very well established mouse tumor models and has used extensively for studying the immunotherapy. So what we find is that compared to the wild type controls, the Mao A knockout mice, the tumor growth is significantly suppressed in both models. And when we look into the tumor infiltrating immune cell, separated, isolated from the knockout mice, compared with those isolated from the wild type mice, we find that the deficient, at the Mao A deficient situation, the immune cells, in particular in this case, the CD8 T cells, which are the important group of immune cell that play a major role in directly attacking tumor cells. We find that for those population of cells, in the absence of a Mao A gene, those cells shows enhanced anti-tumor activity. They produce more cytokines, they produce more cytotoxic molecules. And when we analyze the genome-wide, the signature, of those cells, we find that compared with the cell isolated from the wild type mice, cell isolated from knockout mice contained significantly increased population of the CD8 T cell that show a proliferating and effector cell phenotype. And also they significantly reduced for the population that show exhaustion phenotype. So that all look good, but it comes with one question. When we study the Mao A deficient mice, that every single tissue cell in that mice are deficient of the Mao A gene, right? So our next question is that whether Mao A directly regulate CD8 T cell anti-tumor immunity. In other words, whether Mao A gene is expressed by CD8 T cells and directly play an autonomous role in CD8 T cells, regulate their function. We performed a series of adoptive transfer experiments to address that question. So in one of the experiments, we did the bone marrow transfer. It's a cross bone marrow transfer. We first isolate bone marrow cells either from the wild type or knockout mice and transferred to wild type recipient. And another experiment, we collect the wild type bone marrow, then transfer to the either wild type or knockout recipient. The reason is that in those conditions, at one setup, we confine the Mao A deficiency to immune cells, which are derived from bone marrow cells. At another setup, we confine the Mao A deficiency to the non-immune cells, which is the recipient mouse tissue side. Okay, so the conclusion is the difference for the anti-tumor response only observed when we confine the Mao A deficiency to the immune cells. So that now that we narrow down the regulatory activity for anti-tumor immunity of Mao A within the immune cells. Next question is that there are many immune cells, right? Whether specifically CD8 T cell subject to Mao A regulation. So and third adoptive transfer experiment is that we did some serious breeding setup. We breed a OT1 transgenic mouse into the Mao A knockout background. OT1 is a transgenic mouse recognize the ovalbumin antigen, which engineered into the B16 ova tumor model as a model antigen. Okay, so those cells recognize the tumor, can attack tumor, and belongs to CD8 T cells. So when we directly compare those OT1 T cells either contain the Mao A gene or knockout of the Mao A gene, and we transfer them into the B16 ova tumor-bearing mice, we find that the cells, anti-tumor CD8 T cells, if they are deficient of Mao A, that they significantly improve the ability to suppress the tumor growth. So now that I showed you that we find Mao A gene upregulating tumor-infiltrated immune cells, we confine the deficiency to the immune cells. We find that it's correlated with the tumor suppression phenotype. We confine the gene into the specific tumor anti-tumor CD8 T cell compartment. We find it directly regulate those T cell anti-tumor ability in vivo. And the next question is, how do they do that, right? I just told you Mao A is an enzyme. It's an enzyme break down monoamine substrate and how it regulates a CD8 T cell function. So here's the mechanism study I'm going to show you. So what we did at the beginning is take advantage of the Mao A knockout mice. So we isolate the CD8 T cell from the Mao A knockout mice and compare with them with the CD8 T cell isolated from the wild type mice. So this two type of the CD8 T cells now has one, difference in the genomic, setup, right? So one is totally missing of the Mao A. We compare those cells side by side in the culture, and we characterized the Mao A expression. We find that for the wild type cells, when we stimulate the cell with the antigen, they sharply upregulate the expression of a Mao A, corresponding to the observation that we saw upregulated Mao A expression in tumor infiltrating CD8 T cells. Those cells supposed to see tumor antigen, and that may trigger the upregulation of the Mao A gene. And associated with the Mao A deficiency, we find almost every aspect of the T cell activation has been impacted, including the T cell proliferation, including the upregulation of the activation marker like CD25, and including the secretion of effector cytokine like IL-2 and interferon gamma, and also the production of cytotoxic molecules like granzyme B. So compared to wild type cells, knockout CD8 T cells upregulate the performance on every single aspect of this, like we studied. And to confirm indeed Mao A's work, in an autonomous way to regulate the CD8 T cell function, we also performed a rescue experiment. Okay, the reason is when we directly compare wild type cells with the knockout cells, they are from the different, strain, like wild type and the knockout strain of the mice. Those cells may experience some developmental difference, that may then, translate to their behavior difference later on. So we want to know whether at the mature T cell stage, like if we give back the Mao A gene, to those cells, like is deficient of Mao A, whether we can directly just restore their behavior, to the wild type level, as a evidence to support the direct function of Mao A at regulating the mature CD8 T cells. So what we did that we cloned the Mao A gene. We use a retroviral vector, deliver the gene into the Mao A knockout cells, and we did find that the rescue experiment worked, and the overexpression of Mao A, bring down the overproduction of the cytokine, of the cytotoxic molecule, so together those prove that Mao A directly upregulated by CD8 T cell, and as an autonomous factor can directly regulate the CD8 T cell function. Now we established the correlation that Mao A is an autonomous factor regulating CD8 T cells. But again, what is the detailed molecular mechanism, right? How that really translates to the T cell, behavior. So here is how a T cell responds to antigen stimulation. Okay, in a tumor environment, this antigen will be a tumor antigen. Okay, so antigen stimulate T cell receptor express on a T cell, and the downstream signaling will stimulate the production of all of those, effect molecules, including cytokines, cytotoxic molecules that work together to allow the T cell to kill a target tumor cell. Okay, so what we find is that the TCR stimulation at the same time stimulates the T cell function, also stimulates the expression of Mao A. Okay, and meanwhile, it's also stimulates the production of serotonin. So it upregulates key enzyme called the TPH1 that can convert tryptophan, the substrate, into serotonin. Okay, and meanwhile, CD8 T cell also express surface receptors combined to serotonin. So it's almost like an autocrine loop. Okay, T cell get activated, it secrete serotonin. The serotonin bind to its own receptor by the way, during a response, if many T cells are clustered together in a response, that the secreting serotonin can also work in a paracrine model. But we think the autocrine manner probably is a major way like the T cells utilize serotonin stimulation. And so it's Mao A part serves almost like a negative feedback loop, right? So meanwhile, upregulating serotonin and stimulate serotonin receptor, which joined with the TCR stimulation signal to boost the CD8 T cell response, Mao A can degrade serotonin. Okay, by degrading serotonin, it will drop the stimulatory signal received from serotonin stimulation through the serotonin receptor. And now that if we consider as Mao A as a possible drug target, we can take advantage of the established small molecule, right? The Mao A antidepressant developed for targeting Mao A in brain. And now that it can also target the Mao A expressed in a CD8 T cell. By blocking the Mao A, like we may reduce the degradation of serotonin and promote the accumulation of serotonin and ensure that a helper stimulatory signal keeps sending to the T cell and help the response. Okay, that's why for this aspect, we would like to classify Mao A to this broadly immune checkpoint family, because it works as a negative regulator to block the CD8 T cell reactivity. Okay, so here is the experimental evidence supporting this working model. If we compare the Mao A wild type, Mao A knockout CD8 T cells in the culture, and we measure the serotonin, secreted into the cell culture, we find the accumulation of serotonin is much more significant in the culture with Mao A knockout cells. Okay, and important question is that, is this happening in vivo, right? In a tumor microenvironment? The answer is yes. So here shows the data, like we isolated tumor samples from the tumor-bearing mice. We compared the tumor samples like isolated from wild type or Mao A knockout mice, and we find a significantly increased secreting level of the serotonin in those tumor samples. Okay, and who make those serotonin in the tumor microenvironment is also a important question, right? So we address this question by using some depleting monoclonal antibodies, deplete the different type of the immune cells from the tumor environment. So what we find is the most significant correlation is the CD8 T cell compartment, because when we use depleting antibody deplete the CD8 T cell population, we find that the difference is much more minimized. So great. So now, like we talked about why we are interested in studying Mao A. I showed you like the mechanism study. We find Mao A is induced by antigen stimulation in CD8 T cell, and then work as a negative regulator to regulate the T cell reactivity, right? And then particularly in a tumor microenvironment, this mechanism seems to play over there, and it works through regulating the T cell autocrine serotonin signaling pathway. Then what? Right, so we find this interesting, new target, possible target, whether it's indeed can be, targeted to develop a new version of the cancer immunotherapy. So to do this, we collected a collection of the existing Mao As, and in particular, those has been approved to use clinically. And this one, phenazine, has a treatment name called Nadir, is clinically used at US. We tried all of those Mao A, small molecule inhibitors, and find that they all work very well. Blocking the Mao A activity in CD8 T cells, and by doing that, increase the T cell, reactivity to the antigen response, antigen stimulation. And does it work? Can translate into a anti-tumor benefit, right? So here shows that in a tumor prevention experiment, okay, so we treat the mice, 3 days before giving tumor, and then 3 days later, like we challenge the tumor, challenge the mice with tumor, and we find that all of those Mao A's that we tested, including phenolin, that significantly suppressed the tumor growth. And in a more clinically relevant tumor therapy, setup, we grow tumor first in animal, okay? And then once tumor established, we then start to treat the animal with the Mao A, small molecules. And we similarly, we find that even in the established tumor situation, the Mao A treatment can significantly suppress the tumor growth. And that's associated with the increased anti-tumor reactivity of tumor infiltrating CD8 T cells. It worked for the multiple models, work for the both B16, over melanoma model and the MC38 colon cancer model that we studied. And we, our next question is that, remember we mentioned that one, possible challenge for the current existing ICB, therapy is that even for cancer patients responding to the therapy at the beginning, that the tumor may recur later on when tumor develop the evasion, mechanism to get around that blocking, pathway. So the combination therapy idea is that if you target multiple, it's like the cocktail therapy for AIDS, right? If you target the multiple pathway of anti-tumor immune reaction, that the chance of evasion will be reduced. So that's why we explored the combination therapy. So the underlying hypothesis is because Mao A regulate a very different signaling pathway compared to that used by CTLA4 and the PD-1. So combining the Mao A treatment with anti-PD-1 or anti-CTLA4 treatment that may, bring in the benefit of targeting a separate pathway and give some synergistic effect and possibly, reduce or prevent the immune evasion of tumor. So here shows the experiment that we did. We used the therapeutic model. We tried both the melanoma and the colon cancer model, and we tried the combination therapy, of Mao A treatment with anti-PD-1 treatment. Okay, so like we also included the singular treatment as a comparison arm of the study. So here shows that in this MC38 colon cancer model, which is considered to be a immune sensitive, model, like the Mao A treatment worked almost as well as anti-PD-1 treatment by itself. And a combination of both totally, kind of suppressed and the tumor, but it's already, at a single arm, it's already work really well. Okay, in a more immune resistant model, like B16 over melanoma model, the singular treatment of Mao A shows something like similar to that achieved by the anti-PD-1 treatment. And importantly, the combination of both gave the superior, anti-tumor effector function over here. And that's the only group achieved the clearance of the tumor in our study. Then we ask, it all look good, but we study everything in mice, right? We know that mouse CD8 T cells express Mao A, subject to Mao A regulation, and the targeting Mao A can really change the anti-tumor function of mouse CD8 T cells. Does that happen to human T cells? right? So we go ahead to isolate human CD8 T cells from healthy donor PBMCs, and we stimulate those cells with the CD3, CD28, just give some antigen stimulation to those cells, and we find human CD8 T cells similar to mouse CD8 T cells can significantly upregulate expression of Mao A, of the TCR stimulation. Okay, then the next question is that whether Mao A blockade can really improve the human T cell anti-tumor function in vivo, right? For doing that, we established a xenograft model to study that. So this is how it works. We directly study with the human T cells and the human tumor cells. Okay, the human tumor cells here is a human melanoma cell derived from a line called A375. It overexpresses a matching antigen of a transgenic TCR called the ESO TCR. Okay, so it expresses ESO antigen and the matching HLA-A2 that present antigen to that particular T cell receptor. This T cell receptor is delivered into the human CD8 T cell as a transgene, allow the expression of a tumor responsive TCR and allow that engineered T cells to behave like a tumor specific CD8 T cell, but it's a human cells. And we use a NSG immunodeficient animal model, okay, so that can take the human tumor engraftment, also can take the human immune cell engraftment. So in this model, like we grow the human tumor cells, express this corresponding antigen, okay, on those mice. And then we transferred the engineered human CD8 T cells now being able to recognize the human tumor target after our engineering, after our engineering approach. And then we inject Mao A into those mice. Okay, the question is whether the blocking Mao A in a human CD8 anti-tumor T cells can improve the anti-tumor functionality of that cell. And the answer is yes, in this xenograft model. Okay, so the group that receive the Mao A treatment should significantly suppress tumor growth. Okay, and then when we isolate the human CD8 T cell from the tumor, like from those mice, like we find that the correlation that those T cells indeed should improve the function, like more cytokine production, more cytotoxic molecule, production. Then we go ahead to ask further. I'll say if Mao A indeed, is upregulated in human situation, okay, so in a tumor infiltrating CD8 T cells, whether that's and it's a negative like a regulator over there, right? So like whether, checking the Mao A expression level within a tumor environment from real human like cancer patient samples, whether we can find some correlation of the survivor, of the patient, right? So the underlying, rationale is if Mao A is a negative regulator, then lower expression of Mao A in a tumor, human tumor, may correlate with the survivor advantage of that patient. Okay, so we looked into the publicly available database, like people published the RNA-seq study, like they collect the tumor sample from the different patient cohort. Okay, they posted the data online. We looked into those data, and we focus on studying the Mao A expression, the correlation with Mao A expression, as well as the T cell components inside of that tumor, and then correlated with the patient survivor at the end. I won't go into too much details over here, but you guys are welcome to look into our publication, and it explains that we used a tool called a TIDE, the computational model, like analyze those three elements, Okay, so the survival, patient survivor, linked to the Mao A expression in tumor and linked with the CD8 T cell infiltration in tumor. So the overall conclusion is the Mao A is negatively correlated with the survival advantage that can offered by tumor infiltrating CD8 T cells. So it's then, it's a clinical side of the supporting evidence, to support Mao A is indeed, a factor that may have a correlation with the patient survival. And the same conclusion apply to the multiple tumor type we have studied, including colon cancer, melanoma, lung cancer, cervical cancer, pancreatic cancer, and more. That again, we listed those findings all in the paper. You're welcome to check out. And in particular, a cohort of the melanoma patient already received the anti-PD-1 treatment. That's also a very clear correlation of the Mao A negatively regulate the CD8 T cell function, to offer survival advantage in those patients. So we consider this as a promising piece of the supporting data to show that even in the at the condition of the anti-PD-1 treatment, maybe Mao A, the blockade therapy can offer additional, survival advantage. So combining all of those together, like we proposed this intratumoral Mao A serotonin axis model. I explained to you like what people know about how Mao A work in brain, right? How it regulates the neuronal activity through regulating the serotonin and through regulating the serotonin and controlling the neuronal, signaling transmission, that it regulates the mood and the behavior, change, and therefore, by small molecule targeting Mao A can be used for antidepressant and treat other neuronal disease. And it's a very striking similarity over here. And I also want to bring it to the attention of the audience that the term synapse, it's defined that the two cells interact very closely, form some specialized interaction structure and allowing the signal transmission to happen. And it's a terminology first developed for the neuronal study, like a neuron, synapse like this. And immunology later on borrowed this terminology, and we have something called immune synapse, okay, which specialized characterized as a CD8 T cell right here, that's a tumor cell, for instance, and that's a close interaction of those CD8 T cell and its target tumor cells through interaction through the TCR, recognize tumor antigen, and the assisted by the other co-receptor binding molecules. And at this special synapse, like here, like it's a situation really similar to what happened in the brain, right? So CD8 T cells produce serotonin that can secrete to this cliff over here at the synapse region. It binds to its serotonin receptor and join the TCR stimulation signal, collectively regulate the T cell anti-tumor reactivity. And meanwhile, CD8 T cell also, express Mao A that works as a negative, brake, like it breaks down serotonin, thereby tune down the anti-tumor T cell response. So by blocking Mao A in a CD8 T cell in a tumor microenvironment, then we achieve the accumulation of a higher concentration of serotonin, over the synapse region, and then it can enhance the anti-tumor reactivity of CD8 T cells. So now that we are proposing, as recognizing Mao A is a new molecule belonging broadly to this immune checkpoint, family, and because we can take advantage of existing established small molecule inhibitors for this molecule already, that we can then consider repurposing a Mao A antidepressant for treating cancer, right, for cancer immunotherapy. And another interesting side to consider is that for many cancer patients, especially for those at the terminal stage, like some of them may suffer also from the depression. So maybe targeting Mao A, in this case, may offer dual benefits, both antidepressant and the anti-tumor function for those cancer patients. I promised you we will quickly touch about the other side of the story. We will also find that Mao A, besides directly regulating CD8 T cell anti-tumor activity, also regulate other immune cells participate in the anti-tumor immune response. Another type of important cells called tumor-associated macrophages, another name called the TAMs. Okay, so those comprise the tumor suppressive environment and really play a negative role down to the anti-tumor reaction, like in a tumor microenvironment. We find that Mao A also expressed in those tumor-associated macrophages and also play a negative role. Okay, it promotes those cells to become this more tumor suppressive M2 phenotype of the TAM. And by blocking Mao A, use a small molecule Mao I's, we can convert those TAMs like to a more immune stimulatory subtype, thereby release the suppression function come from TAM. We do find that by regulating the multiple components, like both CD8 T cell and the TAM, Mao A, by giving Mao I and blocking those molecules, Mao A in this overall tumor microenvironment, it can achieve this, multiple, aspect of the regulation and overall improve the anti-tumor immune reaction. I'll stop the talk by acknowledging my labs, in particular, Shirley and Ryan, as I mentioned, two talented postdocs lead the study I just described to you. I would like to thank my many collaborators from Caltech, USC, and the colleague at UCLA. And I would like to acknowledge those funding resource support and make this study I just presented to you possible. With this, I will stop over here. I'm happy to answer any questions you may have. Thank you, Michael.
You can hear me, Lili? That's fantastic. Thank you so much for such a terrific talk. There are several questions in the chat, and there are several questions in the Q&A, and I'll help by being the moderator for this. Okay, so the first question is, does antidepressant serotonin reuptake inhibitors affect CD8 activity? I believe it's in vivo. It says in vitro, but I think the meaning is in vivo. The reason is because many cancer patients take selective serotonin reuptake inhibitors.
Yes, Michael. Yeah, so this is a very good question. Yeah, since we find this serotonin pathway, is important for regulating the anti-tumor reactivity, we actually did explore, the multiple regulatory molecules along the serotonin path, including the reuptake receptor and a few others that also, have been developed as a target for treating depression. So the answer overall is that many of those molecules, besides the Mao A and including the reuptake receptor, those molecules seems all, function. Like if we give to the tumor model, at least in a mouse model, like it can help to boost the anti-tumor immune reaction.
Thanks, Lili. Another question is, will plasmapheresis for the removal of, I believe this means monoamine oxidase A expressing immune cells, help with regression of cancer?
Michael, could you repeat the question? I didn't see it in the chat.
Sure. Yeah, this came through in the Q&A, and it was, will plasmapheresis, so removal, of monoamine oxidase A expressing immune cells help cancer regression?
That's a very good suggestion. I think about it. I would very much like to try it. We haven't done it yet.
Okay. Another one is, will serotonin injection by itself, I assume that means localized, but I'm not sure, help with anti-tumor activity?
We did try that in our study, so it can achieve certain level of the tumor suppression, but by far it's not as significant as targeting Mao A. So the reason behind that, we think, is because Mao A is more like deliver a more localized, effect, right? So like all of those serotonin, autocrine serotonin. So it can be a very high concentration at a local region, and systemically it's a different concentration. So by targeting those molecules, that it may give you a better response. And thinking about how when people treat depression, right? So the antidepressant drug like Mao A, like the other targeting the reuptake receptor, that why don't directly give patient serotonin? So like it has been tried in the past, but other drugs seems to be more like potent.
Thank you. Another question is, since many cancer patients take or may take antidepressants such as monoamine oxidase inhibitors or selective serotonin reuptake inhibitors, are there any studies actually that stratify the activity of checkpoint inhibitor responses to patients who do or don't take those drugs?
Very good question. That is the first line of the clinical correlation study we tried to pursue. But for a very interesting reason, it's very difficult to collect those data. And let me tell you why. Okay, so I did mention that Mao A is the first group of the major antidepressant to be approved, and then its use has been declined like in the past 50 years for two reasons. First is that the appearance of many other like major antidepressant drugs, like the one like targeting reuptake, right? So and another reason is that Mao A, that whole body distribution wide, also expressed in gut, and it's responsible for like degrading the tyramine from a food. And the tyramine is a major component of those dairy products, except in particular aged cheese. So that's a effect called the cheese effect, like associated with the Mao A drug. Is that so people take the Mao A drug if they take a big meal, dinner like with a lot of cheese, and also turkey. Turkey contains a lot of tyramine too, and that may change some the tyramine level, in their body, and that may cause some effect. So, so traditionally associated with giving, treating patient with Mao A, there's a lot of food restriction. People don't really like to follow. That's why the other, antidepressant drug take over. So now it's, I believe it's as a fourth line of the antidepressant for major, depression. And so the patient taking those not that many, and those patients happen to enroll, in a anti-PD-1, treatment clinical trial, the number even lower. So we are still trying, but so far we are not able, haven't able to collect a really meaningful set of data to assess those. But those are extremely interesting questions. I would myself would be very, curious to see what those results will look like.
Thank you. We have a few more. Another question is, can T cells adapt, or do they respond less to chronic monoamine oxidase inhibitor or selective serotonin reuptake inhibitor exposure? Does this effect, I guess, get dampened over time?
Yeah, so that's an interesting question too. So like we are still at an early stage, as at this discovery. So we haven't really tracked the long-term, effect of the treatment. Okay, so yeah, whether that will really cause exhaustion of the cell, maybe too much activation is not good. Those are excellent questions. I mean, the longest in vivo animal study we did is around for like a few months, like 3 months, 4 months. But I guess that probably even longer study, especially looking at the memory compartment, and long-term treatment and the tumor recurrence, all of those will be really necessary to address that question. But those are very important future research directions.
Another question, this might be the second to last, is does monoamine oxidase bind directly to serotonin or other inhibitors of the TAMs? Does it convert TAM2 to TAM1?
Yeah, so like interestingly, even we see the Mao A auto-regulated TAM, it's not through serotonin. Okay, very simple. Macrophage does not make serotonin. Okay, they are lacking the expression of those key enzymes that, for the serotonin synthesis pathway. So that's the first that's the first pathway we explored. I'll say, oh yeah, maybe the similar path also, work for both T cell and the macrophage. It's not. So macrophage is a different, pathway. So it works by, no matter what is the substrate, it will always be degraded and generate the side effect, like the breakdown, kind of components, right? The hydrogen, peroxide, and that belong to the family of ROS, so reactive oxidative, molecule. So in other words, it's responsible by doing that. It regulating the oxidative stress status of a cell. So we find that in macrophages, that is how Mao A regulates the macrophage function. And it's not alone. It's not like macrophages, the single cell like regulated by this pathway. In fact, in brain, okay, Mao A also, can play that role, and it has been implicated for the like Parkinson's disease, like the neuronal toxicity, the accumulative oxidative stress of the neuron really contribute to the disease progression. And Mao A has been used to treat Parkinson's disease because of that regulatory mechanism.
Thank you. I think we have time for one more question. It's a doozy. I think it comes from a clinician. The question is, what hurdles do you think there are for repurposing an antidepressant to activate immunotherapy in cancer?
I would very much, kind of consult with the clinicians, especially clinician scientists in the audience for this. I mean, we are immunologists, like we do the preclinical finding, we can do the preclinical model. We pushed to the xenograft model, to try to directly study human T cells, human tumors. But it's still, we rely on the clinical, side of the participation to develop a new therapy like this, right? So like exact question I want to ask the maybe colleagues, from the audience, right? So anybody interested in following about this, please contact me and we can talk about it.
Well, that's great. Lili, what a tremendous discussion and what a tremendous kickoff to the CTSI speaker seminar series. Thank you so much. Thank you everyone for joining in, and great job. Really interesting work. Thank you so much.
Thank you, Michael. And thank you the audience for your patience. Yeah.
Bye, everybody.